So we just learned about DNA replication, and cells don't replicate their DNA just whenever. There's specific times, and one of these times is during a process called mitotic cell cycling. This video is going to be about mitotic cell cycling. So what is it in the first place? Well, mitotic cell cycling is basically just cell division. What's the goal of that? Well, to just make new cells. And those new cells are going to be genetically identical to each other. So hopefully you're starting to see where DNA replication could play a role in this part right here. So why do cells do this? Why do cells do cell division? Well, it kind of depends. So cells that are unicellular, or organisms that are unicellular, do this process for asexual reproduction. In other words, organisms that are one cell big can make more of themselves just by doing cell division. That's pretty cool. Multicellular organisms, like us, do mitotic cell cycling for other reasons. One of the main reasons is so that we can grow. Did you know that when we grow, it's not because our cells just get bigger and bigger, but we get more cells? Our bodies are made up of trillions of cells. Another reason that we can do mitotic cell cycling is for repair. Have you ever wondered, like when you get cut, how that cut and how your body is able to seal itself up and be almost like new? That's because your body makes new cells to replace those cells that got damaged. And another one is just for replacement of cells, cell turnover. For example, it's said that your skin cells are only about a month old. Your skin cells fall off all the time and are replaced all the time. Well, how are they replaced? By mitotic cell cycling, making genetically identical cells. Now, mitotic cell cycling is a process that's very well understood by scientists, and I want to talk about the different aspects of that process in the diagram down here. Mitotic cell cycling can be broken up into two basic stages, and those stages are interphase and mitosis. But both of those basic phases can be kind of divided up in and of themselves. So, for example, take interphase. Interphase can be divided up into three substages called G1, S, and G2. G1, some people think of that G as standing for growth. And it's true, during this time, the cell size increases from being a smaller cell to a bigger cell because that cell's about to divide. So it'd kind of be nice if the cell gets bigger before it divides so that each cell at the end isn't smaller and smaller and smaller. Also during this time, a cell is making a lot of proteins. And those proteins all have different jobs in the process of cell cycling. Now, I want to draw a cell, and we've been talking about DNA, so it would make sense if I drew DNA in there. So we know that DNA is in the nucleus, so I'm going to draw a nucleus in there, and I'm going to draw 3 chunks of DNA, and this cell's just a bigger version of the previous cell. And so I'm going to draw a nucleus in there with those 3 pieces of DNA. And I want to make sure that we're on the same page with a couple of terms here. And one of those terms is what this part is right here. So the nucleus has its own membrane, and very creatively, that's called the nuclear membrane. And then, of course, we know that the DNA is in the nucleus, but the DNA is organized into chunks or big pieces And those big pieces are called chromosomes. Chromosomes. And that's all one word. I just didn't have enough room to fit it there. All right, so that's G1 of interphase. Next comes S of interphase. And S stands for synthesis. And if you know what synthesis is all about making stuff. And so what's being made here, well, it's DNA synthesis. And another term for DNA synthesis is DNA replication. So this is the stage where DNA replication happens. So how do I draw that? Well, of course, that's going to happen in the nucleus because that's where the DNA is. So let me draw the nuclear membrane, and I'm going to draw our 3 chunks of DNA. And each of these 3 chunks is going to get an exact copy of it. Now, to make that easier to see, what I'm going to do is I'm going to draw the new pieces in a different color. So I'm going to draw those new pieces in blue. And what happens when the copies are made is the 2 copies are stuck together. So it's kind of like the buddy system. And the whole reason why is we're trying to make genetically identical cells. And by keeping those 2 copies stuck together, that makes it easy to organize and sort out. All right, so we went from G1 to S, and now we're going to go from S to G2. Remember what that G stands for? Yep, growth. So the cell is going to grow some more, and the cell is going to make some more proteins and make some more organelles. So when the cell divides, the resulting cells are going to be a good size and have enough stuff to keep on living. Our nucleus isn't going to change any. So the nucleus will still be there. We'll still have our 3 chromosomes that have been doubled. And we call these chromosomes, we call them double-stranded. And these chromosomes are single-stranded chromosomes. All right, so we've gone through interphase. And what has the cell done? The cell's gotten a lot bigger, and the cell has doubled its amount of DNA. And now this mitosis part is where the DNA is going to get sorted out and the cell is actually going to divide. So there are scientific names for each of these regions of mitosis or phases of mitosis, but we're just going to call it the start of mitosis. And this is going to be what happens really early on. And then this is kind of like late in the process. And this is how mitosis is going to end. So start early, late, end. We're not going to worry about the scientific names there. Now, there's a couple of things going on once we start mitosis. I'm going to draw them in, and let's see. I bet you'll be able to see how the drawing is different right away. So the nuclear membrane is there, but the nuclear membrane is starting to disassemble. So nuclear membrane disassembles, and our DNA chromosomes are still in there. And here's the deal, like the nuclear membrane is there for a reason. Keeps the DNA separate from all the other stuff that's going on in the cell so that the DNA is kind of protected from all of those chemical reactions happening in the cytoplasm to decrease the chance of DNA damage or DNA mutations. So the nuclear membrane is a good thing to protect DNA from mutations. But now some things are going to change. These chromosomes are going to have to move around, as we'll see in a second. So we need to get that nuclear membrane out of the way. Now there's kind of like a train tracks or a road system for the chromosomes to move around, and that road system is going to be built right at the start of mitosis. And one road system starts on one side of the cell and one road system starts on the other side of the cell. That has a name called the mitotic spindle or the spindle. So during this start of mitosis, nuclear membrane disassembles and the mitotic spindle forms. Those are the big events. Then, by the time we're still pretty early on in mitosis, we've got a really cool thing that happens. What ends up happening is all of the chromosomes get lined up right in the middle. And the chromosomes are able to get lined up in such an organized way thanks to that spindle. So the spindle actually has pieces that attach to the chromosomes at either side. So the spindle is really important and a really good organizer. So what happens here? The spindle organizes the chromosomes in the middle. Now, if the chromosomes weren't organized, there'd be a good chance that there'd be a mistake here. And our goal is to try to make cells that are genetically identical. And if there was a mistake, there goes our genetically identicalness. So this is a big part to making sure that the cells stay organized. A little bit later on is what happens is these chromosomes start walking down their path of the spindle. So later on, you could see now the cell, instead of being a circle, gets more elongated. These spindle fibers actually get shorter because they get broken down. And it's actually the chromosomes, as the chromosomes walk along the spindle fibers, the chromosomes will actually break down the spindle fibers. So what you can see going on is one identical piece of a chromosome is going to the left side, and the other identical piece of the chromosome is going to the right side. So let's formally write that down here. So what happened was one copy of each chromosome follows the spindle to each side. And now we've got one of these pieces of DNA over here and one over there. One copy of this one over here, one copy of this one over there, one copy and one copy. You can see, hopefully, how we're going to have two genetically identical cells. And that's exactly what happens at the end. Now, at the end, the cell actually divides, and we want to now protect our DNA again because the DNA is done moving around, so the nuclear membrane doesn't have to be out of the way. And I'm going to put the 3 single-stranded chromosomes in that nucleus and the other 3 single-stranded chromosomes in the other nucleus here. So, at the end, we have accomplished our goal. We have 2 genetically identical cells. One of the things that might be misleading here is you see that this one has blue chromosomes and this one has red chromosomes. But remember, these were identical copies that were made back here in S phase by the DNA replication process that we've been studying. So even though they're different colors here, that was just to help us keep track of it. These are 2 genetically identical cells, and that's how mitosis, or mitotic cell cycling, works.